Welcome to the Knowledge Catalog. In this video, we are going to feature another discussion. We're going to have another discussion under the biological sciences. If you are a teacher or a student from the Philippines, you may actually use this as an advanced video lesson or as a reinforcement of your classroom discussions, especially in this new normal education. Particularly, it will be tackling learning competency number 4, which is found in grade 8. It goes as comparing mitosis and meiosis and their role in the cell division cycle. Before we continue, I would like to tell you that this video, the topic that is featured in this video, is not fully connected to this learning competency. But if you want to enrich your students' knowledge of the use of different informatics technologies and sharing and the modalities of sharing information between countries and between scientific institutions, you may actually use this. Okay, so if you want to have a copy of this presentation, you may contact me through my page, facebook.com/knowledge catalog/. Let's begin. Bioinformatics is conceptualizing biology in terms of macromolecules in the sense of physical chemistry and then applying informatics techniques derived from disciplines such as applied mathematics, computer science, and statistics to understand and organize the information associated with these molecules on a large scale. The major center for bioinformatics is the National Center for Biotechnology Information, and as of 2001, the GenBank repository of nucleic acids sequences contains about 11,546,000 entries. The general aim of this branch of genetics is the sharing of information, particularly genes, genetic information that is found in most researches in most parts of the globe. Since there's a really rich level of biodiversity, and sometimes some organisms tend to be even living only in a particular part of the world at a particular kind of ecosystem, it may be a very useful information for the genetic information of those species to be available to those places na hindi sila ma-reach. So for the beginners of sciences such as our students, we may actually explain bioinformatics as a branch of science that deals with the sharing of genetic information from one place to another, and the importance of that is for research purposes. And then, meron na siyang mga derived pang mga importances. Halimbawa, for research on medicine, research on biodiversity levels, and etc. The NCBI has the following data so far, and if I'm not mistaken, this data is from 2017, if not 2018. It is a collaboration. The GenBank is a collaboration between the GenBank of NCBI, the DNA Data Bank of Japan, or DDBJ, and then the European Nucleotide Archive, or ENA, of the European Union. They are exchanging information daily, but the information that they are putting in the database is updated every 2 months, and then it's also being released every 2 months. So the following table, it shows us 3 releases. So release number 3, release number 129, and release number 224 with different dates. So the interval from December 1982 to April 2002 has all of the releases for release number 4 and up until release 128. So ganun siya. As of the third release of the GenBank in December 1982, the bases that were recorded were 680,338, and then the sequences recorded were 606. For the bases, for whole genome shotgun, wala pa siyang laman nun, even for the sequences. However, in the year 2018, the month was February, release was number 224, the GenBank already has 253 billion bases and 207 sequences. Whereas for whole genome shotgun, it already has 2 trillions of bases and then 564 million sequences. The bases and sequences sent to the GenBank and related statistics to them are available online. Anyone can actually have access to them for as long as you have a strong, reliable internet connection and a device that allows you or supports the web page. Information sources for those sequences, for those bases, include the core nucleotide, or it is the main collection, as well as expressed sequence tags, or DBS, that is DB's database, and then genome survey sequences, or DB, database for genome survey sequences, or DBGSS. For you to be able to manipulate or to do bioinformatics techniques here, you have to follow the following steps. Following steps. Okay, so first one is to search for the nucleotide with N-trace nucleotide. In some cases, you may not be actually asked to do the first step because pwedeng ma-provide na kaagad sa'yo yung nucleotide. So, magbablast ka na lang, which is the second step. Okay, so again, the first step is for you to have access to a nucleotide sequence. The second one is to run a BLAST for that sequence. BLAST stands for Basic Local Alignment Search Tool. It aims to find regions of similarity between the sequence searched and other sequences. The sequence that you are searching for is going to show you percentage of similarity. So, gaano ka-similar yung nucleotide na hawak mo from the different nucleotide sequences available online. It will be expressing again that similarity in terms of in terms of percentages. The last step is to search, link, and you can actually download sequences programmatically using the NCBI utilities. You can do that. Now, bioinformatics began with nucleic acid sequences from the GenBank, and then it added protein sequences from the Swiss PROT database. And then it went on to publication of the first complete genome, which is H. influenzae genome. And then it is now conducting different projects concerning gene expression and different projects related to protein structure. All these signify the advent of biology towards information keeping, making computers indispensable. Informatics techniques are more effective with the aid of course, knowledge of computers, since most of data gathering and even data sharing now is done through computers, through information and communication technology. In terms of the aims of bioinformatics, we have been actually stressing the importance of sharing. And I hope that even without us continuing with this part, you already have an idea as to why is bioinformatics important as a branch of genetics. Data organization and access is granted by bioinformatics for researchers. Sharing is done through local networks and even through the internet. By saying local networks, sharing can be done within a particular, say, building that has only one network, and thus yung mga nandoon, they have this common folder or database na pwedeng mabuksan, pwedeng ma-modify, pwedeng makopya ng kahit na sino mang may access dun sa database. Also, pwede rin through the internet, and the network it can be global, it can be worldwide, not just local. Data curation as a task is not actually the end of it. Let us always remember the importance of data when it comes to conducting or taking into effect the steps in your methods. For any researcher, one of the first things that a researcher must be able to do is to gather data. Gathering data can happen by, their own experiments, by conducting their own tests, but some data are not acquired in that way. They can't be accessed in that way. Okay, you can't just produce them, and some of them, even though you can produce them, you still have to test if they are qualified before you even begin with your actual methods. Bioinformatics techniques is going to make that process faster because it will be providing the researcher or different researchers information that they can begin with. Okay, so again, data curation or the collection of data as a task is not just the end of it. Data analysis comes next after data curation. So comparing a sequence with previously characterized sequences is a common task under this aim. And then global comparison can be done to uncover principles across systems and highlight novel features of sequences or of bases. For bioinformatics, hindi lang basta isang uri lang ng data or ng data source ang ipaprocess. Pwedeng hindi lang basta DNA sequence ang i-process. Science now has provided us with sufficient information that, the genetic material can be transcribed, and then the transcribed product can be translated. Pero we are also aware that that process can be reversed. And thus, if you only have proteins, you can actually derive the DNA that has produced that kind of protein, or even just the RNA. You can reverse it one step. Now, the following are the data sources for bioinformatics, yung pinanggagalingan ng mga information ng bioinformatics. The first type of data source is raw DNA sequence. So the topics that are covered by bioinformatics, if you have a raw DNA sequence as a source, is separating coding, non-coding regions, identification of introns and exons, gene product prediction, and then the forensic analysis. Okay, so those are the kinds of topics you can cover for bioinformatics by having raw DNA sequence as a data source. The next one is protein sequence. So if you have a protein sequence, you can do sequence comparison algorithms, multiple sequence alignments algorithms, and identification of conserved sequence motifs. The next one is macromolecular structure. If you have a macromolecular structure, say something that is bigger than a DNA or protein sequence, you can cover the following bioinformatics topics. So secondary and tertiary structure prediction, 3D structural alignment algorithms, protein geometry measurements, surface and volume shape calculations, which are important, intermolecular interactions, molecular simulations, such as force field calculations, molecular movements, and docking predictions. In addition to these sources, we also have genomes, whole genomes. So having that genome, bioinformatics topics you can cover are the characterization of repeats. So yeah, it's important the significance of having a genome as a data source is that you would be able to see what are the sequences that were repeated. And so structural assignments to genes, phylogenetic analysis, genomic scale synthesis, characterization of protein content and metabolic pathways, linkage analysis relating specific genes to diseases. So those are the things that you can do if you have a whole genome. When it comes to gene expression naman, correlating expression patterns and mapping of expression data to sequence, structural and biochemical data are the things that you can do. Other data can be literature or metabolic pathways. So there are digital libraries for automated bibliographical searches. They are not actually biological sources, but they will be providing important bioinformatics information. Knowledge databases of data from literature, as well as for metabolic pathways, we have pathways simulations. So, the data being examined here were already given. So further genomic scale data include biochemical information and metabolic pathways, as it was said, regulatory networks, protein-to-protein interaction data, systematic knockouts of individual genes to test the viability of an organism. Much of the data can be grouped together based on biologically meaningful similarities. When it comes to annotations, the genes can be grouped as in terms of those with particular functions and in terms of those that are according to metabolic pathway to which they belong. Distinct proteins often have comparable sequences. Organisms have multiple copies, while different species have equivalent or similar proteins when they diverge through evolution. The major concern of bioinformatics is information sharing and gathering, as well as, of course, using those information to start researches and to, just satisfy the curious mind. I hope that with this video, you were able to get at least an overview of how important is bioinformatics in the study of cell and molecular biology. If you heard anything in the background, that means I am, a normal person and I don't really have a studio. But I am really hoping that this content has helped you in understanding a very important branch of genetics. The Knowledge Catalog is a brand being developed by yours truly, and I am a high school teacher in the Philippines, and I love developing content I find useful and needed to better my instruction. If you want to support this page, you may leave a like to this video or, a positive comment will do. And if you want to have access or, if you want to be updated with my content, you may subscribe to this channel. See you in the next video.